This webinar should take about an hour. And as I mentioned before, my name is Omar Harb, and I manage the scientific outreach and education for EuPathDB. I'm joined today by Evelina Bashenko, Eve, who is part of the outreach team and will also be covering some aspects of this release. We will cover two things in this release. One is we will mostly focus on new data sets that got integrated and ways you can query these data sets. And we will also then highlight some new features that have been integrated in EuPathDB sites. And of course, if you have any questions, burning questions that you're just not sure about, even if it's not something we're covering, feel free to ask them. And if we don't get a chance to answer them during the webinar, we're more than happy to communicate with you offline and provide an answer for that as well. Okay, so one way that you can find out what is new in our databases is by going to the news section of our sites. Either you can do it on your favorite site. So if you're a user of Plasmodia, you can go to the news section and you'll see here on the left-hand side of your website, you will see that there's a Plasmodia 44 released news item, which you can click on and get more information. Or if you go to EuPathDB itself, then you can see accumulation of all the news items from all of our sites. Each of the news items provides a description of what was added, and you'll notice that many of our databases do not necessarily receive data sets in each of the releases. However, the nice thing is that since all of our databases are built on the same infrastructure, all new features get applied to all of our databases. And as you scroll down, then you will see that there will be databases that receive data sets, which include a listing of these data sets and ways for you to access them. So I'm going to switch for the first portion of this webinar to Eve, who will take over and describe what's new in FungiDB. So,
right.
Okay, and I'm going to go ahead and make you the presenter, Eve, so you can actually show your screen.
All right, thanks, Omar. Hello, everyone, and welcome. My name is Eve, and today I will walk you through briefly over the new genomes that we have recently loaded, as well as demo an RNA-seq query that you can construct using our datasets, as well as discuss some other updates that were relevant to the phenotypic data, as Amar showed a few minutes ago. Amongst the genomes that we have loaded recently, they were 3 genomes, 2 genomes for genome sequence and annotation. One of them was for Cladophillophora immuna and also Rhizophagus irregularis. The third genome, which is just the genome sequence, was for Ameriascus niger. So the Cladophillophora immuna is actually quite an interesting fungus. It's a yeast-like fungi that has a clinical significance, and this particular isolate has been isolated from the Netherlands. While the Rhizophagus irregularis is actually a member of subphylum Gloromycotina that is involved in symbiosis with the roots of most land plants, and changes, for example, in the genetic and the phenotypic components for Rhizophagus species has been linked to plant diversity and ecosystem success. So, for example, one of the type of the searches that you can create with the integrated sequence as annotation is search for various types of genes. I will click on the link here that will redirect me to the genome page. The genome sequence annotation page essentially gives you a brief summary at the top. as to who's the primary contact for this genome and gives you a link out to relevant publication. If you scroll down to the bottom of the screen, you will notice that there is a little bit more information about when the genome was integrated and what was the source, as well as on the bottom, the type of searches that you can create from using these sequences. For example, if you are a researcher that is interested in looking at the transmembrane domain proteins, you may want to instigate a search where you want to identify all transmembrane domain proteins in this particular genome. So I will click on Identify Genes by transmembrane domain count, and the search will automatically essentially run in the background and produce a list of results that will, and I'm being redirected just shortly, but I'll just walk you through really quickly as to what you expect to see. In a little while, you will be able to see that you can set a default search for the transmembrane domain proteins that have either one or more than one domains. And let's see, I have performed the search in the past, just shortly before the call. So I will navigate to my search strategies and see if I can. Here we are. So one of the screens that are still being loaded, and something that I wanted to show you, is how to set up your transmembrane domain count search. You will arrive to this particular screen where the organism of interest is Rhizophagus irregularis, which has been already selected. And for me, I would prefer to leave the number of transmembrane domains at default, which means it will identify any genes that have anywhere between 1 to 99 transmembrane domains. And when you click on Run the step it actually will calculate all the outputs that matches your criteria, and will return a list of genes that match your specifications. For example, here on the right, you will have a column that says, yes, the criteria are met, and you can further look through to see if some of the proteins identified pique your interest. In addition to these type of searches, you can overlap searches with another types of data. For example, if you would like to identify proteins that also have a signal peptides, you can run an additional search by clicking on the Add Step button, navigating to Search for Genes, Protein Targeting and Localization, and look for all genes that are annotated with predicted signal peptide. For example, in this case, when I click on the search, again, I would want to choose Rhizophagus irregularis, and I would want to intersect, in this particular case, the search so that the genes that
Hi, Eve. It sounds like we're losing your sound. At least I'm losing your sound.
are being returned are those that have transmembrane.
Eve, sorry to interrupt. I think
I'll give it just a few seconds to finish up.
Eve, can you hear me?
Here we are. Here is that you can create a multi-step search.
So I think, let me give Eve a second here to correct. I think she's having internet problems. I think.
Sorry, can you hear me now?
Yeah, it's really hard, Eve. You keep cutting out, and I think that's why your websites are not loading for some reason. So there must be some issue with your internet locally at the university. So go ahead and continue, but I will try and send you a message whenever we don't hear your voice. Otherwise, I can demo and show things as you speak.
Okay. Can you still see my screen as I'm reloading right now?
Yeah, I can see it, but it's loading, I think, because of the internet issues.
Yeah, terribly sorry about that. I am connected to the Ethernet, so I'm not quite sure what's happening. Okay, so let me go back to the news and walk you through some of the additional data sets that we have loaded. So in a nutshell, you now can see that you can create searches for specific genes. If the genes are not annotated, and this is rather just a genome sequence that has been integrated, you could set up searches using a specific tool, such as DNA motif tool, where you can look for certain types of motifs within the sequence and identify any interesting sort of hits that way. In addition, we have loaded quite a few transcriptomics data sets, and these span various pathogenic fungi such as Aspergillus, Candida, and Histoplasma. Histoplasma, for example, is a dimorphic fungus that causes respiratory and systemic disease in both mammals and other animals. And this fungus is quite interesting because it has two phases. It has a virulence phase, and it has which is and it also has a non-virulent stage, such as mycelium. For example, if you click on the dataset for the two strains, such as G186A and G217A, it will redirect you to a page where you could compare the transcriptomics data between two strains. And it seems like my page is a little bit slow on loading, but I will navigate to the data set.
So one option, Eve, is you can make me the presenter again, or I can make myself the presenter, and then I will walk through as you speak. I can click as you tell me what to click on. That may work better.
Let's do that.
Okay, so I'm going to switch presenter back to myself. Okay, so as Eve indicated, from the news section, you can click on any of the data sets described and go to the dataset page, which provides additional descriptions of the data set, including a little short description of what the experiment was. But also, as Eve mentioned, in that dataset page, there are links to all the searches that are available to you to investigate this particular data set. The beauty of actually integrating the data into our databases is that it is not necessarily just recapitulating the images from a paper, but rather allowing you to ask questions and discover new things, in addition to coming up with the gene lists that they came up in the paper, but also discovering new things. So go ahead, Eve, let me know what to click on.
Sounds good. And if I can add before we navigate to the RNA-seq fold change, notice that this data is actually for the two different strains, G186A and 217B, while they were aligned to one particular genome. Amara, if you could scroll up, sorry. If you scroll up, you should be able to see the reference genomes. The reference genome against which the datasets were aligned is G27B. This is an important sort of aspect that you need to keep in mind when setting up searches, and why this is useful in this particular case. Note that because both transcriptomic datasets were aligned to the same genome, you are now can compare directly the expression data between two strains, and that opens up a new area for sort of your research, where you can look for the genes that are upregulated in one or the other strain, or both. Amara, if you can go ahead and click on the RNA-seq fold change link, we will navigate to the transcriptomics search window. And here we are. So there's a few things that you need to set up before you can run your search. Notice at the top you have automatically selected that you want to return protein coding genes. You can pick up or down-regulated, or just up or just down-regulated gene. In this case, we will look for up-regulated genes that are which Amar, if you could click on the upregulated genes.
Yes, sorry.
Sorry.
Let me know which ones to click on again. I was answering a user question, which maybe is a good point to interject here. The user was asking, can we re-listen to this webinar? And the answer is yes, we're recording it, and we will make it available via YouTube, but also via a GoToWebinar link. And you should get, anybody who's registered for this webinar will get a link afterwards with the recording of the webinar. Unfortunately, the portions where the voice was cut out, that'll still be cut out, but hopefully you will be able to get what you need out of the webinar. So go ahead and let me know what to do.
Yes, and just to add, if you have any questions, please feel free to contact us and we'll get back to you as soon as possible. So yes, if you could select upregulated genes only and leave fold change at 2.
Which samples should I select?
We will compare mycelia, which is in a virulent stage of histoplasma, to yeast.
Okay, so mycelia to yeast, and do you want upregulated, and you want it at 2-fold only?
Perfect. Yeah, we will leave it 2-fold.
Okay.
Here we are. Once we run a search, essentially we'll return a list of genes that are upregulated in the mycelia. In G217B genome, one of the searches that you can create here is to add a step and run the same search, only instead of G27B, you could use 186 strain. In this particular case, you'll be looking for the genes that are either expressed in both or one or the other strain. As Amar showing, he is clicking on the add step, and then we'll navigate to the bottom of the screen to select fold change again, because we want to run similar searches when creating this type of query. And our reference genome should be mycelia in G186A, which is a non-virulent stage, and the comparison sample will be yeast form. And if we intersect the results, so essentially here we're setting up the same type of searches. We're comparing mycelia to yeast, looking for the upregulated genes in yeast that may be linked to the virulence in this particular strain. So and if we click run step, we will return a list of genes, and because we have chosen the intersect option, it means that only genes that are similarly expressed in both strains are returned. Notice that if you can further click on the edit button, which is within the yellow box when you hover over, you can change the type of the search parameter that you have chosen. You can either look for the genes that are expressed only in G27B or G186. Looks like we probably need to switch the, yes. Yep, excellent. And as you may already imagine, these type of searches open up quite a wide range of new possibilities for you to examine data. You can analyze results, you either go to enrichment, you can additionally add other steps looking for signal prediction peptides, etc. Okay. And as Amar demonstrates, the analyze my experiments window on the bottom, I think I have quite a delay, so hopefully you can see it. You can choose various types of analysis that you can run on the data that are being returned by the search.
So just to clarify again, so what we did here is we asked for genes that are upregulated in yeast in one strain, compared it to, and then intersected those results with genes that are upregulated under the same conditions in a different strain. And this gave us an intersection of 1,139 genes that are in common between both of these strains that are upregulated to the same level. And so depending on what you're asking, this may be a way for you to get sort of a common denominator between all the strains that are operated in yeast compared to mycelia, for example. Did you want me to go ahead and run a Go enrichment analysis?
We could, but I was going to transition to show some of the phenotype updates and the annotation updates that we have had.
Okay, let's go ahead and do that. I can show that in one of my demos. So,
okay, sounds good. So we will show you how to run Go annotations just in a few moments, but for now, I would like to go back to the homepage for the new releases and just talk a little bit more about the phenotype and annotation updates that we have integrated as well. If you scroll down a little bit further down, thank you. There are several sections that we have now available for the community. So first is the phenotype data, and the phenotype data is essentially a manually curated project, in this case for Cryptococcus species, where we curated a paper where 121 putative kinases were deleted, and they were analyzed at 30 distinct in vitro conditions for various types of sort of differentiation defects or growth defects, stress responses, and antifungal resistance. You can read more about the manually created Cryptococcus phenotypes if you click on the link, which will take you to the, Amar, if you could click on the manually curated phenotypes, it will take you to the landing page for this particular data set. And here is a brief explanation of what type of, a brief explanation of what has been changed as far as the history for these genomes. And on the very bottom, it also, as always, offers you a few links to create phenotype searches.
So I'm not sure if I clicked on what you want me to click on. Did you want me to click on the
yes, I just want to demo the phenotype search, so you can navigate.
So manually curated Cryptococcus phenotypes. That's the one.
Yes.
Okay.
Yes. So we could just click on the phenotype search at the bottom, or from the main page.
Okay, just any one of them. The identify genes by phenotype.
Identify genes by phenotype. Yes. Perfect. The first one.
Okay.
Okay, and so here we are. We will look for Cryptococcus neoformans. If you could select from the drop-down menu the second genome, H99. And then from the phenotype qualifier, we will look for decreased. If you click on the second option, we will look for decreased amount.
So there is decreased amount. Okay, so all the way down.
Decreased amount. And then we will look for the phenotype. If you again click on drop-down menu, we will look for capsule organization. So before we run a search, I just want to quickly explain what we're doing here. The text description of the phenotypes that were discovered in this paper have been annotated using various ontologies and controlled vocabulary in such a way that you can create specific searches to identify genes that are linked, in this case, to a decreased capsule formation or organization. Capsule formation is linked to pathogenicity in Cryptococcus species, and as such, decreased formation or absence of capsule is linked to also decreased virulence. So for example, here we will identify any genes. If you clicked on Get Insert, any genes that have a phenotypic description with a decreased capsule organization. And that normally should be about 78 genes, yes, for this particular data set. If you scroll down, you essentially see a list of genes matching your criteria. And this list also tells you the location of the genes as well as their product description. And what is most importantly in this case, you will also notice a column on the right that says PubMed ID. That is essentially a source of the phenotype that has been annotated to this gene. If you're curious as why, for example, AGC, PK, protein kinase have been annotated as decreased capsule formation, you can always click on the PubMed ID link to be redirected to a particular publication associated with it. So now we will switch gears a little bit and navigate.
So Eve, if I can interrupt quickly and answer a question that we received. So
yes,
the question was whether a person can download all the results in a CSV format or any other format, and the answer is yes, and I can show you that quickly right here. So we're looking, let's say this is a list of results, 74 genes that you're interested in downloading. So when you scroll down to the result page, you see here all your results, but you'll notice here that there's a download option, and you can click on download. This will take you to our download page, which allows you to customize what you want to download. So there are various formats depending on what you need. For example, you can select a tab delimited Excel format, and then you can select here from any option, any information we have about the list of genes. You can select whatever you want. And when you can show it in the browser, download it as a text file or as an Excel file. But let's say I leave this as the default selections here, and I click on Get Answer. This shows me in the browser what you get, but this can be downloaded obviously as an Excel file. Another very important and I think very useful download feature, that is the FASTA format. So this is basically just a text file of your a structured text file of your sequence. And so, for example, if I click on FASTA, you will get some options here. You can simply just say, well, I just want to get the protein sequence, of all of these genes, and that's fine. You click on this, and you get the sequence, and it will dump a list of all the protein sequences in one shot. What's really cool, though, I think, is that if you're interested in looking at different parts of the genes, but you want to get that information all together in one shot, you can, for example, for these 74 genes, let's say you're interested in knowing if there's something in their upstream region of the start sites of all of these genes, and you want to take that sequence and reanalyze it somewhere else in your favorite program to identify, let's say, transcription factor binding sites in the upstream regions. Instead of having to go in and do this for each sequence individually, you can go in and select that you want to download the genomic sequence, and now you can configure, well, what part of the genomic sequence for all 74 genes do you want to download? So I can, for example, change this, and I say I want to select anything that is beginning at the translation start site. I want it to be, and depending on if I want upstream, you select minus. And there's a description at the bottom of the page here that will describe how you can run this query. But let's say I select 500 nucleotides, and I can change this now to the translation start site because I'm only interested in the upstream regions of all of these genes. If I keep it at 0, this is going to be the ATG, the A of the ATG of your methionine codon. If I don't want to include that, I can just make this into a -1. Or if I actually make this into a +2, this should give me, each one of my sequences should end in the ATG, right? And so I always like to do this as a test to make sure it's downloading the right stuff. And so when you click answer, in one shot, it gives you the upstream region for all of these 74 genes, and you'll see here that each one is ending in an ATG, which means that this query is working appropriately. So I can go back and modify this and make it into a -1 if you're worried. But now you can take the sequence and take it into another program, for example, for identifying motifs. And another important point here in configuring this page, you notice that the FASTA headers. So if I get the sequence again, you notice that the FASTA headers are quite long. There's a lot of information here, and this sometimes is a problem for some programs. And so what we have is for you an option to simply say, well, I only want the gene ID to be in the FASTA header. And now you'll get the same sequence, but instead of having a long FASTA header, you just have the gene ID. So you can take this and do whatever you want with it. But the nice thing about this is that you're doing this for all genes at the same time. Okay, Eve, go ahead.
Okay, perfect. Thank you. So let's go back to the news release main page, and I will just finish up with the annotation updates for fungal species. If you scroll down just a little bit more, we will visits the updated functional annotation and annotations that have been loaded from the Gene Ontology Consortium. So essentially, when we talk about annotation updates or gene ontology, we talk about the knowledge that have been accumulated about the function of genes. And gene ontology normally describes this knowledge of the biological domain with respect to three aspects. There's a molecular function, biological process, and cellular component. The examples of molecular functions, for example, could be catalytic activity. The examples for the biological process could be such as signal transduction or DNA repair. And if we're interested in cellular component, for example, we can look at any functional annotations that are relevant to a ribosome or a mitochondria, etc. So we're continuously working on improving functional annotations for some of our genomes, and summary of annotation updates are always available as a link out within the within the news pages. So Amar, if you could click on the summary of annotation updates, notice that in this release we have updated records for Cryptococcus neoformans H99 and JC21. And if you click on the summary, it will essentially take you to a Google Doc, which first gives you a summary of all updates that went in, such as there were updates to the names, synonyms, Go terms, and PubMed IDs, as well as comments. And if you're interested in looking a little bit further in as to what type of changes were made per each strain or per each genome, you can click on individual tabs at the bottom. For example, if you click on C. neoformans H99 log, you will get a log of all updates for this particular genome in release 44. And as you can see, some of the Go terms were added, etc. So speaking about code terms, Amar, if you could go back to the news release page.
Okay.
I will just mention the Go terms that were updated for the Candida. So notice that the Go term annotations were pulled from the Gene Ontology Consortium for essentially 7 Candida genomes, and these are now integrated as Go annotations. And as Amar will show you later, Go annotations can come quite handy when you're trying to enrich the list of genes that you obtained as a result of your search and see whether you have some sort of enrichment that could be interested for molecular function of the genes or cellular component or both. So at this point, we probably will switch gears, and I will give the screen and the lead back to Amar, who will talk to you about new genomes in other databases as well. Thanks, Amar.
Okay, thanks, Eve. And sorry again about the technical difficulties, but hopefully the message went across, and I'll try and demo some of the features again in using the other genomic sites that we have. And so, as we indicated in the beginning, each of the databases each of the databases gets a news release, but then of course the ones that have data, you will see in the data section there will be additional information. And so I'm looking now at the news section of GeardiaDB. And as you can see here, that there were a couple of new genomes added. These are unannotated, and as Eve indicated, when you reload an unannotated genome, obviously you cannot search for genes, but you can certainly look for open reading frames and do a blast against it or look for motifs, as Eve indicated. For Giardia, we also loaded a transcriptomic data set from Stefan Zver's group, and that's again available for you here. I won't demo this right now. It's a similar, the searches are all, since we're all built on the same infrastructure, it's the same type of search in terms of running fold change or differential expression and so forth. But I will demo this in one of our other databases again, just to make sure it is clear. ToxoDB also received some new data. So I'm looking again at the ToxoDB news release page. And you'll notice here that the data set that got loaded was restriction fragment length polymorphism data. And so depending on what you do and what you work on, this may be data that you're not familiar with, but in many of the genotyping world where you're trying to identify isolates in the environment or from various animals, for example, Often one way that people type the isolates is using restriction fragment length polymorphism. It's a very inexpensive way to actually type your strains, and we have ways for you to query this data based on the genotype. So these are the different fragments that are mapped on a gel essentially, and so you can go in and select that you want, for example, the SAG1 protein to be of the type 2, and then you can modify this as you wish, and then run the answer, and it'll give you back all the isolates that met this criteria. And in this case, we're not returning genes, we're returning isolates. And so these are all isolates that came from, in this case, it looks like it came from a cat, and it had this particular genotype. I selected it, it should have a type 2 SAG protein genotype, and so you'll notice all of them, the genotypes will, at least one of the genotypes that are indicated should be this type 2 SAG1. Another database that got new data this release is Pyro Plasma DB. So Pyro Plasma DB includes both Babesia and Theileria genomes. For this release, there was a proteomics data set, quantitative proteomics data set for Theileria parva sporozoites. It's only one condition, and so there's no comparison you're going to be doing here, but what we do offer for you is a way for you to identify proteins based on their percent expression. So essentially we rank all the proteins based on their expression values and rank them, and so now you can go in and say, well, I want to find all genes from Theileria parva sporozoites from this proteomics data set that were highly expressed, let's say between the 80 and 100th percentile of the search of the expression level. And once you get answer, you will get a list of proteins, in this case 250 genes that met that criteria. Those are from this proteomics experiment. Of course, you have to be aware that the proteomics experiment will not be comprehensive. You won't get results from all genes, just as is the nature of the technique. But at least from the proteins they got, these 250 were one of the most highly expressed proteins in the sporozoid condition. You can do various analyses on these. So for example, I can quickly click on this genome view option. It will paint the location of the genes across the chromosomes. And so this is a quick way. It's not really necessarily statistical, but it at least gives you a way to quickly see if there's any kind of clustering of genes in particular regions. So of course, if you're interested in seeing if your genes are clustered in the telomeric ends, we would see some clear bias here if this were the case. The other option is you can analyze your results and do various analyses on them, Go enrichment, metabolic pathway enrichment, or word enrichment. All of these are running a statistical test, a Fisher's exact test, to determine if something in your gene list is enriched over the background, which is the genome. For gene ontology, we're using the coterms that Eve was talking about. So those coterms are very important because they give a unified way of describing various aspects of biology, including cellular component, molecular function, biological process. And so you can select one of these processes and ask, and of course you can change your p-value cutoff if you want, and then submit this. And that question you're asking, well, is there anything enriched in my set compared to the background? And you'll notice here you get Go terms that are enriched. In this case, there's this intracellular non-membrane bounded organelle. It looks like it's a very high enrichment score. Of course, you have to investigate these further and ask, do these make sense? Am I getting anything interesting here, or is there some artifact in the analysis or the data? Can go back and look at the analyses. So one question is asked, why do we have this word enrichment? And the reason for having this word enrichment is that this simply what it's doing, it's looking at the names of the genes, the names of the proteins, and asking, based on the names, is there anything enriched? So if I submit this, we might get a bunch of junk, basically, something that's like putative or hypothetical, right? That's enriched, and that's of course a possibility. And this is exactly what's happening here. I'm getting putative is highly enriched. This means that many of the genes in my list are actually annotated as putative. But often what happens is you can have genes that have names, but do not have gene families, for example, that have names like var genes or other types of variable genes in many of the organisms we study, but that do not have a Go term. And so those will be lost in your Go term enrichment. So it's often good to do this as a sanity check to make sure that you're not missing some important genes in your analysis. Okay, so let's go back up here. I'm going to go to TriTripDB. So TriTripDB also received some data sets, but also a number of annotation changes. So depending, something we didn't mention before, but depending on the organisms we have, there is a support for curatorial effort. And so this includes some of the fungi and Candida plastida, and also there's annotation efforts on Plasmodium species. And so anything that gets updated and we integrate into the database gets mentioned here. I won't go through this again. It's going to be exactly what you saw in FungiDB, except for Trypanosoma, at least the Trypanosoma that were updated. But you can quickly see here that there have been a number of changes since the last release that were updated. Many of the changes come from user comments. And as I scroll down here in the news, you'll see here that they're curated user comments. And you'll see that in L. mexicana, there were 31 genes that had user comments on them that became curated. In other words, that what was provided in the user comment by the users, by people like you. So you can go to any gene page that you know something about that gene, whether if it's something that's published or something that you know from your work. Even if, for example, you tried to knock a gene out and it always failed, which could indicate that it's essential, but that information is quite useful. And you can go ahead to any gene page and add that user comment. Curators then will look at these comments, and if it affects the annotation, they will use that comment and change the annotation. Of course, they may send you an email and ask you additional questions and so forth. So you'll see that there were, for L. mexicana, there were 31 genes that had user comments that affected the annotation, and for T. brucei 927, there were 32 genes that had the same effect.
Sorry,
yes.
And I just wanted to quickly add that in addition to the GFF files that Omar showed you at the top, you can always download Go annotation files, GAF files, and all of those will be available if you navigate to the Downloads menu at the top. That is our gray menu, and navigate to the current release folder that has a particular genome that you're interested in.
Right, a good point, Eve. So what I did quickly is I clicked on Downloads and Data Files, and you'll notice here there's a folder for, let me go back one page. When you come to this Downloads directory, you will see here that we have a folder for every single of our releases, right? So if you were ever interested in knowing, well, I wonder what was different between release 1 of TriTripDB in a particular genome compared to the current release, you can always download the GFF file, the status of the genome during that release, and compare it to the current release. The current release is always at the top. If I click on this, it'll take you to the current release. You'll see all the lists of genomes, and I can quickly navigate then to any of the genomes of interest. So we can go ahead and pick this L. mexicana genome, and you'll notice here there are different folders, each one indicating what files you should expect to see in there. So for example, if you were interested in the Go annotation files, you click on this GAF file or GAF folder, and it gives you a GAF file that you can download. And if you wanted to get the GFF file because you want to do some other analyses, then you can click on GFF here and get the data. And this will provide you with the GFF file. And the same thing for the FASTA file. If you just want the entire sequence, you just click on the FASTA folder, and then you will notice that there'll be multiple types of FASTA files available to you. Obviously, one for the annotated coding sequences, one for the proteins, one for the transcripts, and the genome itself. And then we do prediction of open reading frames. So if you're trying to do some other types of analyses, you may want to get these. Often we get the question, well, I'm running a proteomics experiment, and I would like to get a file that I can use as a database for searching my peptide profiles. And this is where you get this. This is a FASTA file of all the annotated proteins. And so you can download this and provide it to the proteomics facility where they will use it as a database for research. You could actually do the same thing with the ORFs. As you notice, this file size is much bigger, and that's because there are many more ORFs predicted. And so if you're trying to discover proteins that map to open reading frames that are not necessarily in genes, that have not been annotated as genes, that's a nice way to then discover new proteins. Okay, so So with the interest of time, I'm going to jump to PlasmoDB here, and I'll demo some more of the searches. So I'm going to PlasmoDB, the current release, and this displays the data sets again, as we've been describing. There were 2 RNA sequence data sets that were loaded, including a red blood cell stage developmental cycle transcriptome. And then we have this one, which I think is quite exciting. This is a sporozoid transcriptome in different microenvironments. If I click on this, it takes you to the dataset page, which gives you a description of the dataset, a link to the paper, so you can read more about this paper to understand what this experiment was. But essentially what they did is they took Plasmodium vivax sporozoids and grew them under different microenvironment media conditions, so in vitro culture conditions that mimicked various environments, including the salivary glands, but also mimicked the blood stages. And so if you're interested in then identifying genes that are differentially regulated in different stages, you can, for example, as you've indicated, I can go to the fold change query. And now when I run the search, I can ask, for example, I want to find all genes that are upregulated. Let's change this to something like tenfold. And let's compare this Schneider's media plus BSA, which at 0 hours, so this is basically sporozoids. And let's say I want to compare it to RPMI plus BSA. This is supposed to mimic blood exposure to blood, basically. And so now when I run this search, and you click on get answer, you'll see here, in my case, I got 0 genes. So you may say, oh, shoot, there's something broken. And the answer is no, there's nothing broken. It just means that the criteria I selected returned no genes. And so that's totally fine. So you can go in and go back and edit this and say, well, let me revise this. Is there, maybe my fold change was too high, so let me change this back to 2. And so that's a nice way of working with the data and to see if you can discover genes that are potentially differentially regulated, and also it doesn't really cost you anything to run this search and get 0 results, and then now you know the answer, and then you can go back and modify it and change the parameters. As you can see, here's a list of the results. As I scroll to the right, you'll notice here that you get a graph, and this represents the entire experiment. So you can see where this gene is changing in expression in the different conditions. And it looks like in any condition where there is, well, most conditions where there is Schneider's media used, there's upregulation in fold change. Okay, let me go back. So one thing I wanted to highlight also about the search, hopefully you notice this graph here on the right. So this graph is supposed to give you a visual cue as to whether you're selecting the right criteria and whether this makes sense to you. Is this what you were trying to find? So in this case, I had only 2, I'm comparing 2 samples to each other. So I said, I want to find genes that are upregulated in this 0-hour Schneider's plus BSA, and you get a red dot up here, and I want it to be upregulated by, in this case, tenfold above the RPMI plus BSA. So you get 2 dots here, and the tenfold is between these 2 samples. If I change this to 2, then what happens is this graph just dynamically shows you twofold. But now if I add additional samples, so let's say I want to compare, I want to compare all the Schneider media compared to all the media that has RPMI. So I'm just going to go in here and select anything that has the word RPMI in it. So I selected all the RPMI ones. And you'll notice here that you get let's stop here, it's probably enough. And so you see here, there are a bunch of dots here. And now I can select here a bunch of the ones that include the word Schneider in them. Let's just select these here. So you see here, I'm comparing 4 samples to each other. And you'll notice as soon as you select more than 1 sample, you get an additional criteria here that you can play around with. Right now, it's selected as average. So I'm saying that I want the twofold difference to be between the average of the expression values for these 4 genes and these 4 genes. There are 4 samples, sorry. You could change this and say, well, I want to select the difference. Let's see, this is not cooperating with me for some reason. So this is supposed to show you an additional drop-down menu, which is not showing. So there might be a bug in the in this particular search page. So we'll follow up on that. So that's another maybe a good learning point here for everybody on the call. If you ever come across a page and you see something that doesn't look right, don't assume that it's your fault and you just don't understand what's going on. There's quite a likelihood that maybe there's a bug in here. And so I would encourage everybody to click on the Contact Us link and then send us a comment or a question and say, hey, I tried to run this search and I can't find this particular option. What's going on? And so that's something to keep in mind. Okay, so what happened is when I changed this, for some reason it wasn't picking up the menu item, so I changed it to upregulated again. Now you'll notice here you have other options. So I can change this. For example, I want to compare, I want this twofold difference to be between the maximum expression value from these 4 samples, and I could change this now to minimum. That twofold needs to be the difference between the maximum expression value for these 4 samples compared to the minimal expression value for these 4 samples, right? And so this graph then gives you a visual cue, well, is this what I want to find? And now when I run the search, again, the answer may be 0, so let's find out. And the answer is 0. So, and of course, the more samples you select and the stricter you make your parameters, the less likely you will get a result. So that's something just to keep in mind, that there are very many different ways you can play around with the data and ask questions about the data. Okay, so let me go back to homepage here. One more data set I wanted to highlight is we loaded some ChIP-seq data, which is only available as JBrowse tracks. And so these were a particular histone marks that mark actively transcribed regions of the genome. And again, there's a lot of information here that allows you to understand what this data set is about. But what you can do is you can go to the genome browser in Plasmodia, and as a note, right now we have 2 genome browsers that are active. We have the Gmod genome browser, which is the old genome browser, which we are phasing out slowly, and the new one, JBrowse, which is a more modern browser and a bit more responsive and faster. And so I'm showing you here just the JBrowse for Plasmodia. And I can go in here and select tracks, and you'll notice here there's a bewildering number of tracks, so it's a bit crazy, right? But one thing you can do easily is start typing a search up here, and it'll filter out of these 935 data tracks, it'll filter down to the track that you're interested in. So in my case, this is an ATAC-seq data, and so very likely the data set actually has the word ATA-seq in it. And so I'm just started filtering now, ATAC, and you'll notice here that I got all the samples from this particular experiment. I can either select all of them to turn them on, and as I'm doing that, you'll notice here in the background it started loading them, and I can go back to my browser, and now you'll notice here that I have all the tracks loaded for this particular data set. And so let me scroll back to the top. So here are my genes. This is a density plot. This is all the data compressed into one density plot, and you'll see here the darker areas shows areas where there are peaks, and you can see that these coincide with the peaks that are below here for the ATA-seq data set. Okay, so let me go back here, and I want to demo a couple of features. So we only have 4 more minutes in this webinar, so there are 2 features that we wanted to demo. One of them is as many of you already know, potentially we have a mechanism for you to analyze your own data in using the Galaxy interface, and I'll quickly show this. So I'm just going to go to ToxoDB. Let's go back up here, and let's go back to the home page. And you'll notice here that there is a section called Analyze My Experiment. And so if I click on Analyze My Experiment, it'll ask you to log in and do different things. So logging in is required because this uses essentially resources that will be linked to your account. So this is a private database, but as I'm logging in, this will load the Galaxy interface. And this will be my Galaxy workspace. So any data sets that I've analyzed before will be there. So you have to accept this. Basically, we're not responsible for storing your data forever. So just keep that in mind. If you ever analyze data with any of these resources, actually, you have to be careful that that's not going to be a permanent storage for your data. So always make sure you download your data that you analyzed if you're interested. So this is the Galaxy interface. If you're familiar with Galaxy, this should be old to you. If you're not, there are a number of tutorials out there that are available to you, and we're happy to help you as well and walk you through any of these types of searches. We have a number of pre-configured workflows which allow you, for example, to analyze and compare two samples to each other and run a differential expression analysis. And this is what I did right here on the right. So this is in this case, this was a Plasmodium experiment where I compared samples by differential expression. But what we have is a feature that allows you to export the data back into EuPathDB and to start asking questions about it. And we have EuPathDB export tools up here on the left, and you can choose from any of them. So for example, this case, the RNA sequence to EuPathDB export. And remember, this is all private. And what you do is you just fill up the information here. And a new feature we added for this release is that we also now allow you to transfer strand-specific data back into EuPathDB. So the data will be mapped in a strand-specific manner, which is quite useful. And for the next release, we'll be developing a search for you that allows you then to search and ask, for example, find me all genes that have an increase in reverse strand expression or antisense expression compared to forward expression when I compare this sample to that sample. If I go back to Plasmodb, for example, and log in, just to show you what this looks like. And so since I already logged in ToxoDB, once I click on login, it automatically logs me in because we use the same username and passwords. So even though you're somebody who works, for example, on Cryptosporidium and you always use CryptoDB and you have a login there, that same login will work on any of the databases. So if I go to my dataset section, any dataset that I exported from Galaxy will appear in this section. And so this allows me to quickly then take a look at them and ask questions about them. So depending on how many datasets you have listed in here, this page may take a few seconds to load, but here are my different datasets that I have loaded. I can, for example, let's pick, doesn't matter which one, let's pick this one here. And so this case takes me to my dataset page. So this is a private dataset page. You'll notice here that I shared this data with a couple of people. I can click on this share and put their username that they use for logging into the account to Plasmodb, and this data becomes automatically shared with them. So this is a nice collaborative way to work with other people in your group. And what you'll notice here is that there's now a search that's available to you. So I can click on RNA sequence on my user data set. And when I click on this, this is just the same search we were running before, but now it is with your data. And it's also private. So nobody else can see it except for you or people you've shared this data with. So now I can start asking, are any of my genes upregulated? In this case, these are female versus male gametocytes. And I can ask again, I can change this to maximum compared to minimum, right? And so twofold is fine. I can run this search. And so this will now return, in a familiar way, it returns my list of genes that were upregulated in my female gametocytes compared to my male gametocytes. If I scroll down here, you will notice that there's a graph as well. This is a custom graph that's dynamically generated based on your data. And so you can quickly see what's going on here. You can run Go enrichment analysis and all the various tools. Essentially, everything is available to you based on what's in the databases, but now you can run it on your own data. One thing to note, and that's a question we get often, and I'd like to mention this, is that if you have your own list of genes, so for example, if you ran an experiment in your lab and you have a list of 100 genes or 200 genes, gene IDs that you're interested in integrating so you can ask questions about them, you can do that easily. So I'm just going to go back to the home page to show you where the search is. So if I go back to the home page and I go under annotation, curation, and identifiers, and now when I click on this, it shows you these three searches. I can click on gene IDs. So now under gene IDs, I can paste the list of gene IDs. So if I have 1,000 gene IDs, I can just paste them in here, click on get answer. And what will happen is I will get a, I don't have a list of IDs right now, but if I paste them in here and click on get answer, what will happen is I will get a step in a search strategy with my gene IDs that are uploaded. And now you can start asking questions about them. And so that's a very powerful tool that I would encourage you to do. So the last thing that we wanted to demo quickly is to show you the that we've integrated Synteny into JBrowse. And Eve, would you like me to just continue demoing this because of your internet issues, or did you want to show this?
Yes, please go ahead. I have tested and I'm still having trouble loading.
Okay, no problem. So let me go and let's select any one of these genes. So let's go ahead and select this first gene. And let's say the question is, from an evolutionary perspective, you're wondering, well, does this gene, is it synthetic across various Plasmodium species, and is the region synthetic? And so what I can do is I can click on, go to the JBrowse genome browser right here. This is already available in GBrowse. It's always been there, but the new thing is that we've now integrated it in JBrowse. And again, if you're familiar with GBrowse, it's very slow, and so hopefully this would be a welcome change. And so if I go to the orthology and Synteny section, you'll notice here there's a comparative genomics track. I can click on this, and now if I go back here, it should load a section that shows you the Synteny. So these are basically, we align all the genomes to each other, the nucleotide sequence, and then we shade all genes that are orthologous of each other. And so you can now quickly take a look, and obviously this region is highly synthetic across all the Plasmodium species. And so that's a nice quick way to do that. The beauty of having it in JBrowse is that this is very dynamic, so I can scroll to the right. It takes a few seconds to load, but then it loads, and that's actually very fast if you consider the amount of data that's being sort of transmitted and displayed on your screen. Okay, so I think this is everything we wanted to cover. I'm going to give everybody maybe another minute or so to consider if they have any questions they want to ask. So feel free to raise your hand or type your question, and then we'll go ahead and end this webinar. And of course, if you have questions that are not necessarily anything we covered in the webinar, feel free to ask them now. I'm happy to cover them, or we can do that offline. Okay, it looks like we don't have any more questions. So thanks everybody for attending. And as I mentioned, this webinar has been recorded, and we will make that available to you. And obviously anybody who could not join the webinar, they will get to see it. All right, thank you so much, and see you next time. All right, bye-bye.
Bye.